Welcome to Biology Made Easy. Today we are looking at differences between mitosis and meiosis. It will be very useful if you understand chromosome structure and then homologous chromosomes. Please check my lessons on these. Now, mitosis and meiosis differences. Mitosis, one nuclear division. Meiosis, two nuclear divisions. We'll explain. Mitosis, one cycle of prophase, metaphase, anaphase, telophase. In meiosis, you have two cycles. You have prophase 1, metaphase 1, prophase 2. So you have two cycles of prophase, metaphase, anaphase, telophase. The result of mitosis is two identical nuclei or cells for growth, repair of tissues, and asexual reproduction. Meiosis results in four genetically different haploid cells for sexual reproduction and also to produce variation within the species. Now, prophase in mitosis is very short in comparison with a long prophase 1 in meiosis. Now, in prophase 1 in meiosis, there is synapses, bivalent formation, chiasmata, and crossing over. We will explain. Now, here we have mitosis, meiosis. This is early prophase. We have 4 chromosomes, 2 homologous chromosomes. This is maternal, this is paternal, if you like. We have just used symbols to label these as homologous chromosomes. This is also another homologous chromosomes. So we have 2 sets of chromosomes, 4 chromosomes here. In meiosis, early prophase is the same as we discussed in our last lesson on similarities. Very early prophase, we have the same. The chromosomes are thin. By middle prophase, you could see that each chromosome is actually made up of 2 chromatids with a centromere. So you have this as this one as this chromosome as this one as this. The same in meiosis. However, meiosis, by middle of prophase 1, the homologous chromosomes have paired up. Look at A pairing up with its small a here, B pairing up. There's no such pairing up in mitosis. And the pairing process is the synapses. Each pair is a bivalent. So this is a bivalent, this is a bivalent. Now, another thing that happens in meiosis is that these pairs undergo crossing over. So, explaining chiasmata formation and crossing over, we have this homologous chromosome, this chromosome and this chromosome are homologous. Now, there's going to be chiasma formation. It's just the point. You see this chromosome and this chromosome? This ones are sister chromatids, this ones are sister chromatids. This and this are non-sister chromatids of this homologous chromosome. So we have a break between 2 non-sister chromatids. Between this and this, they come together, they lie together, even though they will be repelling. But then the point where they are held together is called the chiasma, and it's at the same point that it will break and they will exchange parts. And that is the crossing over. So the point here, chiasma will break, this will join this will join this as we have. And any genes on this part will be exchanged. A gene here will get into here and a gene here will get. That's how genes are mixed up through crossing over between non-sister chromatids of homologous chromosomes. So the crossing over is between homologous chromosomes. It will not take place between one chromosome and the non-homologous, and it's between non-sister chromatids. That is what happens in meiosis. It doesn't happen in mitosis. Then at this metaphase at mitosis, you see the homologous chromosomes, they are arranged in a single line at the equator of the spindle. Here's the spindle, here's the spindle. So at the equator of the spindle, this chromosome is here, its homologous pair is here, this chromosome is here, its homologous is here. So the chromosomes are arranged in a single row at the equator of the spindle. But in metaphase 1 in meiosis, the bivalents pair. So they form two rows at the equator. So this chromosome is here and its pair is down here. They pair up at the equator. This chromosome is here, its homologous is here. They pair up at the equator. So they form two rows at the equator at metaphase 1, but only one row. We will come back to show that in metaphase 2, the chromosomes arrange themselves in a single line. We will explain. Then we come to anaphase. Anaphase in mitosis, sister chromatids separate. Anaphase 1 in meiosis, homologous chromosomes separate. Now here is metaphase, here is anaphase. At anaphase, the centromere holding the sister chromatids break. So the centromere here will break and this chromatid will be pulled up and this one down. So in mitosis at anaphase, the centromere breaks and sister chromatids are pulled to opposite poles of the cell. In anaphase 1, rather, the homologous chromosomes that had lined up at metaphase 1, they separate. So this chromosome goes up, this one comes down. So at metaphase 1, there is what is called independent assortment of chromosomes start. At anaphase 1, independent assortment of chromosomes happens. We'll explain independent assortment in our next lesson. In anaphase in mitosis, sister chromatids separate. In anaphase 1 in meiosis, homologous chromosomes separate. Good. Then you have telophase. Telophase in mitosis, the chromatids that separate, they've reached the poles of the cell. And the chromatids, they thin up and look exactly like the thin chromatin we started with. So the nucleus divides and you have two nuclei with 4 chromatin, just like we started with. And so at telophase in mitosis, you have 2 identical cells with the same chromosome numbers as we started with. Meanwhile, at telophase 1 in meiosis, look here, we have 2 nuclei form, each of them with half the number of chromosomes that we started with. We started with 4, now we have 2 in each. So telophase 1, we have a haploid number of chromosomes. Now we are going to go to prophase 2, and then we'll compare what happens in here as we have shown here. Prophase 2, the major difference is that the spindle is formed at right angles to the former spindle. So look at the spindle and look at the spindle here. In mitosis, metaphase, the chromosomes line up at the equator. At metaphase 1 in meiosis, the bivalents pair up at the equator in 2 rows. But in metaphase 2, look at these chromosomes. In prophase 2, metaphase 2, they line up, the chromosomes line up in a single line at the equator. Then the difference between anaphase in mitosis, anaphase 1 in meiosis, and anaphase 2 in meiosis is that in anaphase 1, homologous chromosomes separate. But anaphase 2 in meiosis is just like anaphase in mitosis. Anaphase 2, the centromere breaks and the sister chromatids. You see, this centromere will break, this chromatid will be here, and that chromatid will be there. Centromere breaks, this chromatid is here, and this chromatid is here. Anaphase 2 is just like anaphase in mitosis. Then, after anaphase 2, we are going to go to telophase 2. And in telophase 2, the chromosomes or chromatids, they've arrived at the opposite poles and the nucleus divides, and you end up by having 4 nuclei, 1, 2, 3, 4. Each of them with a haploid number. So note the difference between telophase in mitosis and telophase 1 and telophase 2. By telophase 2, the 4 nuclei are formed, and each nuclei contains half the number of chromosomes that we started with. For instance, in this nucleus, you have this chromosome and this chromosome. You see? This and that here. And the other one, you have this and this chromosome, this and that. And in these, you have a mixture of the 2 sister chromatids. That is because of the chiasma formation and crossing over. So, in telophase 2, you have 4 haploid cells. Well, if the lesson has been useful, please give us a thumb and do subscribe. Thank you very much. Bye-bye.